Welcome to the student presentation week 2025. Myself Samruddhi Khairkar, student of B Pharmacy 5th Sem from Patil Institute of Pharmacy Talegaon. So, my presentation is on RUTA. So, the contents are introduction, synonyms, biological sources, geographical sources, cultivation and collection, macroscopic characters, microscopic characters, chemical constituents, extraction and isolation, solubility, uses, and marketed products. Introduction. RUTA falls under the class of flavonoid glycoside. It was first isolated in the year 1860 from buckwheat. It was also isolated from RUTA graveolens in 1941. Now, the synonyms of RUTA: common rue, garden rue, herb of grace, and fringe rue. Biological source. RUTA consists of dried aerial parts of RUTA graveolens Linn. This plant is small evergreen shrub cultivated in temperate region. Family is Rutaceae. Geographical source of RUTA. It is native to southern Europe, especially Italy, Spain, Greece and Balkan regions, and also widely cultivated in India, North Africa and West Asia. Cultivation and collection of RUTA. In cultivation of RUTA, there are many factors. First one is soil requirement. It prefers well-drained sandy or loamy soils, and soil should be rich in organic matter with good aeration. pH should be ranges from 6.5 to 7.5. Second is climate. Thrives well in subtropical and temperate climates. Requires moderate sunlight. Too much heat can reduce growth, and tolerant to drought but sensitive to frost. Third one is propagation. Propagated mainly by seeds or stem cuttings. Seeds are sown in nurseries during spring. Transplanting is done after seedlings reach 15 to 20 cm in height. Fourth, planting. Spacing should be 45 to 60 cm apart. Pits are fitted with farmyard manure before transplanting. Fifth, irrigation and manuring. Requires moderate watering, avoid waterlogging, well decomposed farmyard manure, compost added during growth. Periodic weedings and hoeing needed. Now, the collection of RUTA. First, part used. Leaves and aerial parts are collected from medicinal purposes. Second, harvesting time. Leaves are harvested twice a year, first in summer before flowering and second in autumn. Now, third one is method of collection. Mature leaves are hand-picked when they are green and aromatic. Avoid collecting during flowering stage as oil content reduces. Fourth is post-harvest handling. Stored in airtight containers away from light and moisture. Macroscopic characters of RUTA. Drug should be dried aerial parts, mainly leaves and sometimes flowering tops. Leaf type is bipinnate or tripinnate. Shape is broad, oval to oblong leaflets. Surface is smooth without hairs. Odor is strong, unpleasant and aromatic. Margin is irregularly lobed. Microscopic characters of RUTA. Leaf, transverse section of lamina. Epidermis, single layered, covered with cuticle, both upper and lower surfaces. Trichomes present, mostly glandular trichomes. Mesophyll, differentiated into palisade and spongy parenchyma. Oil glands numerous, scattered in mesophyll containing volatile oil. Vascular bundles, collateral, located centrally in midrib, surrounded by parenchymatous bundle sheet. Last, calcium oxalate crystals, present as small clusters in mesophyll and parenchyma cells. Chemical constituents of RUTA. The primary chemical constituent of RUTA species include alkaloids such as acridones, phenolines, and furoquinolines. Flavonoids like rutin, coumarins and furanocoumarins such as bergaptan and xanthotoxin. Volatile oils containing ketones and phenolic acids. These compounds are responsible for RUTA's diverse pharmacological activities, including antimicrobial, antioxidant, and neuroprotective effects. Now, next one is extraction and isolation. Extraction. Extraction of RUTA involves using solvent like subcritical water, subcritical ethyl alcohol, and subcritical carbon dioxide, or organic solvents like methanol and ethanol to isolate active compounds such as furanocoumarins, alkaloids, and flavonoids from the plant's roots or aerial parts. Now, the isolation of RUTA. The isolation of compounds from RUTA graveolens involves extracting powdered plant material, typically leaves or seeds, using solvents like ethanol or methanol, followed by liquid-liquid extraction and various chromatographic techniques such as silica gel column chromatography and HPLC to separate and purify specific compounds like rutin and various alkaloids. Now, the solubility of RUTA. RUTA is generally insoluble in water but soluble in alcohol and organic solvents. Chemical test for RUTA. Chemical test for RUTA species which are rich in flavonoids and other compounds include Shinoda test and alkaline reagent test for detecting flavonoids. First, Shinoda test for flavonoids. Preparation. Mix the RUTA sample in 95% ethanol to prepare an ethanolic extract. Reaction. To the ethanolic extract, add a few drops of concentrated hydrochloric acid and 4 pieces of magnesium ribbon. Observation. A color change to orange, red, pink or purple indicates the presence of flavonoids. Uses of RUTA. It is used as nervine, antispasmodic, carminative, analgesic, capillary straightening, antihelminthic, antifungal, antimicrobial and anti-inflammatory. Here are some marketed products of RUTA. Now, let's move to the acknowledgement. So, I express my heartfelt gratitude to Dr. Nilesh Banarasi sir, our esteemed guide, for his continuous support, motivation and valuable guidance. I am thankful to respected Principal Sir, Director Sir, all the faculty members and non-teaching staff for their encouragement and assistance. Special thanks to my friends and classmates for their cooperation and moral support. Thank you. Have a nice day.